What is RNAi? RNAi is a process by which small RNA molecules and proteins interact with mRNA to precisely regulate translation. This process has been observed in many different organisms, suggesting it is a common evolutionary trait. So how does it work? There are two main types of RNAi. siRNA stands for short interfering RNAs. These are not endogenous. They are usually a result of viral or bacterial infection or another method of introduction of foreign DNA. miRNA stands for microRNAs. These are endogenous sequences in the genome that can be expressed in response to a signal. For miRNAs, we start off with the desired gene. This gene is a long sequence that is largely complementary to itself. This is transcribed into pre-miRNA, which forms a hairpin structure. This pre-miRNA hairpin is then cleaved and phosphorylated by an enzyme called DROSHA, which removes a portion of the stem of the hairpin. This leaves a small overhang and a 5' phosphate group. The pre-miRNA is transported into the cytoplasm, where it binds with high specificity to dicer, which then binds to the double-stranded region of the hairpin, measuring roughly 22 nucleotides from the terminus, and cleaves it to form a roughly 22 nucleotide long double-stranded RNA segment. After this 22 nucleotide long double-stranded RNA molecule is formed, it is incorporated into an Argonaute complex. This binds tightly to one RNA strand, while the other complementary strand is degraded. This 22 long single-stranded sequence on the Argonaute protein is used as a probe to find any complementary mRNA sequences. One of two things can happen. If the probe binds with perfect complementarity, the mRNA is cleaved and degraded. If the probe binds with imperfect complementarity, the complex binds and prevents translation. siRNAs are a little bit different. Like we said earlier, siRNAs are not endogenous. They tend to form long, linear, double-stranded RNA molecules. In humans, processing is performed by a RISC, otherwise known as RNA-inducing silencing complex. Three proteins, dicer, TRBP, and Argonaute 2, associate with each other spontaneously. This binds double-stranded RNA, dicing it into roughly 22 nucleotide long double-stranded molecules, just like miRNA. This molecule is directly loaded onto Argonaute 2, which cleaves the passenger strand, forming a functional RISC. Generally, these RISCs target mRNA sequences that are 100% complementary to the guide strand. Once complementarity is established, the complex cleaves the RNA strand, allowing for suppression. Why is this important? When RNA interference was discovered in the late 90s and early 2000s, researchers found that they could use this as a useful tool to determine gene function, heredity, expression, and a host of other applications. This mechanism has actually been employed in many of our foods. Some GMO crops are thought to have developed a pseudo-immune system, allowing for protection against infection through degradation of foreign RNA. RNA interference is still an area of active research, with large amounts of the precise mechanism still unknown. However, there is much promise in this field, with possible applications in medicine, industry, and biotechnology. Neat, huh?